Hello, everyone. Welcome in. We're glad to see you out tonight and on Zoom. My name is Morgan Gifford, and I am the STEM Education Manager here at the National Atomic Testing Museum. On behalf of the museum staff and the Nevada Test Site Historical Foundation, I would like to welcome you all out to our distinguished lecture featuring Dr. Audrey Williams. During her presentation, Dr. Williams will be providing an overview of the Forensic Science Center, including their history and current events or current efforts to respond to and deter the use of weapons of mass destruction, as well as their work in support of the Organization for the Prohibition of Chemical Weapons. Dr. Williams is happy to answer audience questions at the conclusion of her talk. If you do have a question, we would ask that you please wait until a staff member has brought you a microphone. This will allow everyone attending remotely to hear your question. And for those of you who are watching remotely, we would please ask that you write your question in the Zoom chat box, and we will be sure to ask that question for you. It is now my pleasure to tell you a little more about our speaker. Dr. Audrey Williams is the director of the Forensic Science Center at Lawrence Livermore National Laboratory. There, she leads a team of scientists to understand, prepare for, characterize, and respond to a variety of threats, including chemical, biological, radiological, nuclear, and explosive scenarios. Now, please join me in welcoming Dr. Audrey Williams.
Hey, thank you for that introduction, and thank you for the invitation to speak to you guys today. This is my first time at the museum, so I'm excited to be here. I'm excited to be able to see it, and I'm excited to share a little bit about our work at the Forensic Science Center with you all. So I will warn you up front, she did mention that we do chemical, biological, radiological, nuclear, and explosive work. I'm going to be talking a lot about chemical threats today, to give you a little flavor of a different type of WMD than I think the museum covers. But hopefully by the end of my presentation, you'll see that the threats, and there's some common threads that weave through these WMD threats, no matter which type it is. So let's see. I point to the right thing. There we go. So I'm from the Forensic Science Center at Lawrence Livermore National Laboratory. As I'm sure you're aware, Lawrence Livermore is one of the 17 Department of Energy national laboratories around the nation. Many of the national labs are run by the Office of Science. There's 3 that are run by the NNSA, the National Nuclear Security Administration. Livermore is one of those. We're located in the San Francisco Bay Area, about 45 miles east of San Francisco. And you can see from looking at the map, there's actually 4 national labs in the Bay Area. So we have a lot of great colleagues in the region that we can work with on some of these issues as well. Livermore itself was established back in 1952. We have a little over 8,000 employees now, and our facility is about 1 square mile. You see here the aerial photo. Having 8,000 people in that 1 square mile has its good and bad sides. When you need a new laboratory, there's not much space, so that can be a downside. But when you need to find an expert in something, some area of science that is not your specialty, you have 8,000 colleagues that you can find right here in our own backyard, which is really really great. So it really has more of an academic feel to it. There's a lot of different expertise that we can call upon and really try to do our best work to support the nation. We're operated by Lawrence Livermore National Security, which is an LLC. The primary members of the LLC are listed here. University of California, unsurprisingly, being located in California, is one of the major members of that LLC. The lab's mission is listed here overall, and it really is to strengthen national security. We do that through world-class science, technology, and engineering, as you can see by the 8,000 employees that we do have. There's 4 main mission thrusts at the laboratory. The first is nuclear deterrence. As that issue has evolved since the laboratory's founding back in 1952, it's really moved more now into a stockpile stewardship and lifetime extension support role in that regard. Threat preparedness and response covers a lot of the other threats, the chemical threat, the biological threat. There's been a lot of activity, unsurprisingly, since the COVID-19 pandemic began in this area. What have we learned there? How can we use that to be better prepared for the next biological threat or the next pandemic that may come our way? We also involve a lot of intelligence-informed decision-making in this area as well. So we do have a intelligence program on site to make sure that we are aware of the latest and greatest threats that we may be facing as a nation. The next is climate and energy resilience. The laboratory does a lot of work in modeling climate change, trying to develop ways to abate climate change, looking at energy stability, whether that's carbon capture, whether that's looking at the stability of infrastructure around the nation. And then the final one is integrated deterrence and competition. And that is really focusing on emerging threats and a multi-domain battlefield, and what do we need to do to be prepared in that area. No matter which of these missions we're focusing on at the time, it really relies on our scientific backbone, which is science, engineering, and computations. In all of these areas, we really do see those disciplines working hand in hand to make sure that we can address these threats and really bring the best solutions to the table as we can. So the Forensic Science Center, we have our own mission, which is listed here as well. But we consider ourselves a national security asset that has a bifurcated mission. On one hand, we're conducting innovative research. And on the other hand, we're performing operationally relevant forensic science analyses. Those two missions really do span the whole WMD threat space: chemical, biological, radiological, nuclear, and explosives. And on one hand, as we're doing research and development, we may be developing new methods, analyzing new threats, learning about how materials interact with different surfaces. And we really are becoming experts and have a body of knowledge that really covers those materials. And so when an operational sample comes in, whether it's from law enforcement, whether it's from DOD or another partner, we can then almost hit the pause button on that R&D and take that state-of-the-art technology we've been developing, take those people that have been working on these threats day in and day out, and now we address that operational sample. So we can look for those novel threats. We can use these detection methods we've developed. We have an understanding of the materials now, how they interact with different matrices, whether it's a piece of tile or carpet or a soil. And so we can really understand that problem. We intentionally keep those two missions working simultaneously and using the same people because they really do kind of play off of each other. So we can develop R&D, apply it to an operational sample, but as we're analyzing that sample, then we can identify some gaps. We can say, gosh, this family of compounds is really hard to extract from a humic soil, or gosh, there doesn't seem to be, this compound that we're seeing in these samples doesn't seem to be in any of the analytical libraries. And we can take those gaps and feed them back over to the R&D side of the house. So sometimes that means we see a gap and we say, gosh, it would be great if we could understand how this type of chemical interacts with this kind of soil. We write a research proposal, we find an agency that wants to fund it, we get the funding, and we start, a research project that may last a year. Sometimes that sample is so urgent that we say, ooh, here's a gap. We need to have an answer on this sample in 48 hours. Do the R&D really quickly, figure out what the problem is, create the solution, and immediately apply it back to that same sample. So we kind of work on those timelines. Can be 1 or 2 days, or they could be 1 or 2 years. And it's that ability to kind of work and weave through those two missions, I think, that really makes what we do unique. The 4 circles here indicates the main focus areas of our work. I mentioned, threat agent characterization is a big part of it. We do try to stay on top of the threat. What is, the latest and greatest chemicals that are being used or considered as hazards? What are the, biological agents that seem to be of a high threat? We look at those materials. We look at how they degrade. We look at how they interact with surfaces. We look at measures that could be used to defeat them, whether that's an environmental cleanup, if there were to be a chemical exposure, how do we determine that a facility is safe to re-enter, or it could be, developing actual countermeasures for individuals. So if someone's exposed to an agent, what can we treat them with to speed their recovery? The next one is the real world sample analysis. That's where we do the sample analysis. One unique thing about our group too is that since we do work with chem, bio, rad, nuke, and explosives, we have the facility to actually work safely with all of those materials. So we are the only place in the nation where you could theoretically take a chemical warfare agent, a biological agent, rad nuke material, and an explosive, stir it all up into a single sample. Not that this would ever happen, but if it did, we have the safety protocols in place and the staff training in place that we could work with that sample to exploit all of those different threats. International engagement and treaty support. I'll be telling you a lot today about the work that we do with the Organization for the Prohibition of Chemical Weapons, which is more on the international treaty front. And we do a lot of support for at-risk nations as well, trying to help them build their analytical capabilities so that they too could respond to the use of WMD. And then the final one is a little bit of a stretch, I think, for the WMD world, but we are looking at developing some novel approaches to traditional forensics. So in traditional forensics, you may think, fingerprints, hair, all of these things that the National Academy of Science put out a report back in 2009 calling for a need to really strengthen forensic science. There was a lot of criticism that a lot of traditional forensic techniques are very subjective. It's an expert that's testifying. I've been doing this for 35 years and my expert opinion, this hair looks like it's consistent with this hair and that they're from the same source, but that's very hard to quantify. It's very hard to reproduce. And so I'll be telling you a little bit about the work we've done there to try to bring some more objective and quantitative techniques into the traditional forensic world. Oops, wrong way. So here's a little timeline of the FSC. We were founded back in 1991. And at that time, with the way that the world was changing, our first director, who was at that time just a scientist at the laboratory, looked around and said, gosh, we've got all this great analytical equipment. And we could really be serving law enforcement or the intelligence community or the US government at large in a better way with these tools and capabilities that we have. And that's really how the FSC was born, really trying to bring the capability and the knowledge base that existed at the lab to those additional communities. There's a few early investigations that I highlighted on here. The Unabomb investigation was one of the first large, more federal investigations that we were a part of in the mid-90s. I have the Angel of Death on here, which was a California local case. But it's an interesting one because I do think it's a good example of how our missions work together. So this man here, the Angel of Death, Efron Saldivar, was a respiratory therapist. And he was killing his patients because his workload was too heavy and he wanted to lighten his responsibilities. And so he had confessed and then recanted his confession. And so what the investigators decided to do is they pulled all the patients that had died at the medical center that he worked at that had died while he was on shift, went through all the records, determined which ones had been buried and not cremated. They ended up exhuming 20 of the bodies and came to us and said, hey, do you have a method to detect Pavilon in patients in exhumed tissue? And we said, no, but we can make one. And that's really kind of how this operational and R&D mixture really kind of brought to bear on the sample. So we were able then to use animal tissue, develop a method, find out what the right markers are, how do you extract it, how do you prep those samples, develop a method, validate the method, and then actually turn to the samples of exhumed tissue from the case, and we were able to detect Pavilon in 6 of the patients, 6 of the victims. And when they went back and reviewed the medical records, there was no legitimate dosing with Pavilon in any of those patients. And so that data was critical in convicting the angel of death. He was convicted for 6 consecutive life sentences and is still in jail down in Southern California. So that's really a case where, normally we don't get involved in local law enforcement issues like that, but it was something where our unique capabilities could really help advance the knowledge and capabilities of the community. There's a nuclear flavored one here down on the bottom, labeled smuggled HEU. This was a highly enriched uranium sample that was seized. Someone was crossing the Bulgarian border and had this in the trunk of their car. I include this to highlight, on the nuclear forensic side of it, we could then, we could take that little ampule of uranium. You can't see it in the picture, but there's an ampule containing uranium in there. We can analyze that uranium. We can determine the enrichment level. We can look at the chronometry to figure out when it was last reprocessed, compare to databases, and come up with when we think it was last reprocessed, plus or minus 50 days, what facility we think it comes from. And that kind of covers the nuclear forensic side of it. But we also take all of the rest of this into account. So you see this yellow material in there. That's a wax material that the ampule was sitting in. We were able to analyze that wax, determine that composition. There's a dye in that wax that's actually what's giving it that bright yellow color. It's a barium chromate based dye. That's a carcinogen, so it's not used in Western nations. That wax is also very absorbent. So we could look at the organic materials that had absorbed there. We could potentially link samples based on the environment they'd been in if they had absorbed the same compounds. We were able to look at the lead container that the whole thing is in. So that's more databases that exist. We can look at the isotopics of the lead. We can look at the impurities there, figure out where that lead material came. And you can't see the top of the container here, but on the top of the container, there's a sticker with some writing on it. We were able to speciate the tree that the wood pulp that made the paper came from. And that tree only grows in a specific part of the world, right? So yes, it's a uranium sample. And yes, the nuclear forensic information is very important, but there's a lot of more traditional forensics that comes in a sample like that we really like to pull all those threads together. OPCW, I'll be talking to you about a lot today. That's the Organization for the Prohibition of Chemical Weapons. We work closely with them as a designated laboratory. We've been a partner lab for the FBI. They have a formal kind of hub and spoke relationship with many laboratories that have some of the specialized capability. So we've had that relationship now for many years. And then on the right side, just one more of kind of the nuclear flavor here. If you remember, there was the issue at the waste isolation processing plant, the WIP, back in 2014, when a barrel burped and had a radiological release at the WIP, which shut down the movement of transuranic material to the WIP for several years. We were involved in the team investigating what happened, where the radiological release came from, and doing the forensic analysis of samples that could be collected safely to determine, what happened, and trying to figure out now, how can those materials, the rest of the materials from that same batch, how can those be moved safely, and what needs to be done to understand that the safety of those materials and how likely it would be for another one to burp or have a kind of exothermic release in the same way. I put a few of our R&D activities over here on the right. You'll hear a little more about some of the proteomics work and our novel biomarker discovery, so I won't dwell on those here. So no matter what threat we're looking at, we really do try to look at the whole threat timeline. Oftentimes forensics is really kind of considered in the response phase. There's been an incident and now we're trying to collect samples and figure out what happened. But we really think that there's a lot that goes along that whole timeline where forensics can bring a lot to the table. So we look at acquisition of materials. If you're talking, a chemical agent, then we're talking about the reactants, so the starting materials that were used to synthesize something. We look at synthesis. So what are the different ways that you can make something? What are the different ways you can acquire it? Are there ways that we can distinguish those from one another? And distribution and smuggling, we think about detection in many cases. So how can we screen for hazardous materials to ensure that they're not coming across the border, or that a package contains what we say it, or what the label says it contains. Use and release really focuses on detection, identifying the material in a timely fashion so that we can provide actionable information to the people that need it. And then the postmortem forensics is really looking at after that kind of where forensic is traditionally thought of as playing a part, looking at exposure signatures. How do we tell what someone was exposed to? How do we clean up a scene? How do we really respond and take that information back to assist law enforcement? So this kind of detect, prevent, and respond is kind of the overarching mission, but really what we're trying to do is anticipate it. We don't want to be doing all of that discovery and all of that learning when an event has already occurred. We would like to anticipate what the possible threats could be, what the possible ramifications could be, and make sure that we already have that solid foundation so that if it does happen, we can respond quickly and efficiently. So I'm going to talk to you a little bit about chemical warfare agents. So I thought I'd give you a little primer since it is a little different than the nuclear topics that you see in the museum. The chemical warfare agents can loosely be defined as these 6 categories here. There's choking agents like phosgene that almost can create like a drowning sensation in the lung. Blood agents like cyanide, which will affect your body's ability to use oxygen. There's blister agents like sulfur mustard, which creates some blisters like you would see here, really affect the mucous membranes. So your eyes, your nose, your lungs again. Nerve agents would be like sarin or VX. These cause overstimulation of your nervous system by inhibiting acetylcholinesterase and other cholinesterases in the body so that your nerves continue to fire. There's incapacitating agents. These are things like fentanyl that really have that kind of incapacitating effect that can knock you out. And then finally, there's riot control agents. This would be like tear gas or mace. The interesting thing about riot control agents, although they are considered chemical warfare agents, they do have some legal purposes, particularly in law enforcement use. So that's one that kind of is in that gray area that it depends on why you're using it, whether it would fall into the category of a chemical weapon. So when you think about the history of chemical weapons and where they came from and when they've been used, really the first large-scale use of a chemical weapon was in World War I. When there's a photo here of soldiers releasing chlorine gas on the battlefield in Belgium. This is a large-scale release, really using it as a weapon. That was then followed by more use of chlorine, phosgene, even sulfur mustard at the time. But after World War I, the Geneva Protocol went into place in 1925, and that really banned the use of chemical gases in warfare. So it was the start of trying to put some restrictions on what these types of materials could be used for. Surprisingly, there wasn't a lot of or really any acknowledged use of chemical weapons during World War II on the battlefield. Of course, the concentration camps is a different story. But there's many different explanations for why that is. Part of it, I think, is thought to be the fear of, if we have these materials, what does the other side have? And it's kind of this idea of the kind of chemical arms race, not wanting to be a victim of a hazardous material if you were retaliated against. But certainly there was more chemical weapons developed in the 30s and 40s. That's when all the G agents, so like sarin or soman, tabun, were all developed, as well as VX, both in Germany as well as the UK for VX. The next major use that was seen of chemical weapons was in the Iran-Iraq War, when sulfur mustard was used by the Iraqis in Iran. And it was right around that time when they really started trying to broaden that Geneva Protocol. And what turned into the Chemical Weapons Convention negotiations started occurring with some of the conferences on disarmament and trying to control the use of some of these chemicals. So that was initially established back in 1980, and it took a long time to get all the negotiations and figure out exactly what the Chemical Weapons Convention was going to be and what it was going to say. But in 1997, the Chemical Weapons Convention, CWC, went into force. This is a great accomplishment. It took a lot of time and energy, and there was, I think, many times when they thought it was just going to fall through and not get there. The nice thing about the Chemical Weapons Convention that was different from the Geneva Protocol is that the Geneva Protocol made it illegal to use chemical weapons in warfare, but it didn't say anything about making them or researching them or stockpiling them. It was really just about the use. But the CWC really did ban the use, the research, the development, the stockpiling, all of those activities with the materials. And also required their destruction. So the existing stockpiles needed to be destroyed to be a signatory to the convention as well. There's currently, it started out slow with countries signing one after another, but as of today, there's 193 state parties that are signatories to the convention. So it really has widespread application. Part of the CWC as well, had to define what exactly does it cover? Where are those lines that we can say something is a chemical weapon or related to a chemical weapon and what is not? And that's what you see on the right side of the slide here. So the CWC set up what they call schedules. So there's Schedule 1, Schedule 2, and Schedule 3 compounds. Schedule 1 compounds are the chemical agents or primary starting materials that have no other legitimate use except to make or be a chemical weapon. So those are the ones like sarin or VX that really have no other purpose to exist. Schedule 2, again, are either chemical weapons or something that's used in the production. They have limited small scale applications for other purposes. So something like BZ, which is an incapacitant, or thiodiglycol, which can be used as a starting material for making sulfur mustard, but also can be used for other chemical syntheses as well. And then Schedule 3 is really getting into chemicals that, yes, they can be used as a CW or to make a CW, but they have a lot of other purposes as well. So things like phosgene, PCl3, which is used can be used in making sulfur mustard, as well as a lot of other chemical syntheses as well. So it tries to tier the chemicals as to how certain you can be that they're related to CW. So when the treaty first went into effect, it was pretty straightforward. Since the CWC did require destruction of stockpiles and did have this very kind of chemistry focused definitions of what was illegal and what was not, they created the Organization for the Prohibition of Chemical Weapons, which is sort of the enforcing arm of the CWC. So that's an organization that's housed in The Hague in the Netherlands. It's an international body that's responsible for ensuring that people that have signed the treaty are following through on those commitments. So making sure that the stockpiles are being destroyed, making sure that no facilities are making chemical weapons. If they say they're making a pesticide, are they actually making pesticide or are they making CW? So there's a big inspection arm that goes with OPCW's responsibilities. And that's what you can see in the pictures here. And some of these are actually doing observing the destruction of materials. You can see them unloading some devices here and looking at production plants and making sure that everything is done in accordance with the treaty. So that was 1997. OPCW was very successful at getting all that destruction of the materials started and ensuring that signatories were following through on their commitments. But really, if you look at the world today, and really over the last decade, we've seen a lot more use of chemical weapons. Starting really with Syria, in the Syrian civil war, there was sarin used, as well as ISIS used sulfur mustard in Iraq and Syria as well. So there's a lot of materials a lot of instances where these materials were being used. A little bit later, it sort of took a turn that chemical materials started to be used in assassinations. So it was no longer a battlefield issue. Now, when we look at Kim Jong-nam being assassinated in the airport in Malaysia, that was a very targeted attack. It was one person attacked with VX. You look at the Skripals in the UK, where a Novichok agent was used. The attack was on the Skripals, but of course there was 2 other victims that were exposed to materials that were discarded. And then Alexei Navalny also being poisoned with a Novichok agent. So now that it's really that shift from a battlefield issue to an assassination issue. And that's something that the CWC never really considered. What these issues resulted in, particularly the use of these Novichok agents, is the first modification to the CWC in its history. So it had been in effect since 1997. Then in 2019, they finally added a new class of compounds to the treaty, which were these Novichok type agents. They also added carbamates, which is shown here on the right. It's another type of chemical that there was some patent literature that showed these materials had been researched. And then one specific compound was also added to the schedule. So there was four additions that were made here to the CWC. This was a big deal because they had to get the international community to all agree on what should be added, what shouldn't be added, to make sure that we're using that treaty in the right way as an international community. So with all of this use of these chemicals, the responsibility falls on OPCW to determine if they are being used and if they're being destroyed properly. And so OPCW relies on a designated laboratory network to do that chemical analysis. As I said, there's 193 state parties that are signatories of the CWC. There's typically about 22 to 24 countries that have a designated laboratory. So if you look at this map here on the slide, the countries that are shown in dark blue are countries that have designated laboratories. So there's relatively good coverage over the world. There's many laboratories that can help support the OPCW when they do collect samples. And so I do want to draw attention to a few things on the map here. You see the green stars. Those are countries that have at least two designated laboratories. So the United States has more than 2, Russia has more than 2, China has 2, and South Korea has 2. So those are the 4 nations that have 2 laboratories. The reason the United States has 2 laboratories is that when we signed the treaty, it's almost like they put a little asterisk next to their name, which is condition 18, which says if a sample is ever collected inside of the United States, it will not be sent outside of the United States for analysis. They did that to protect intellectual property issues. If there was a facility in the US that was accused of making CW, OPCW comes in, collects a sample, sends it to 2 other countries to analyze. They aren't making chemical weapons, but now you've given those proprietary formulas to other nations and haven't protected that company's best interests. So OPCW's mandate requires that 2 independent laboratories analyze any sample that they collect. And so that's why the United States has to have at least 2 designated laboratories so that we can fulfill OPCW's requirement as well as protect US IP. So if you're talking about environmental samples, which was really when the CWC was created, that was really what they were targeting. That could be things that were taken in a laboratory, a sample that was found in a hood or a waste container, maybe a soil sample collected outside of a facility. They don't have to be environmental per se. That was how OPCW first started. And so for those types of samples, the 2 designated US laboratories are the Forensic Science Center at LLNL, as well as DevCom CBC, which is the Army Combatant Command Development Command Chemical and Biological Center. It's a mouthful. They've changed their name many times. Everyone refers to it as ECBC. It used to be the Edgewood Chemical and Biological Center. So we're the 2 that are certified for environmental samples. Several years ago, OPCW started to broaden their reach and started looking at biomedical samples as well. So individuals that may have been exposed So in the biomedical samples, they're looking at blood or urine or tissue. Those types of methods, analytical methods, are a little bit different. And so some laboratories operate in the biomedical space and some just don't. So in that scenario, in the United States, the Forensic Science Center and DevCom CBC are both also certified there. But the CDC, the Center for Disease Control and Prevention, as well as the U.S. Army's Institute of Chemical Defense also plays. So the United States actually has 4 designated labs for biomedical samples. And the last thing that OPCW has really been pushing on lately is biotoxins. So biotoxins are in that in-between space between chemistry and biology. Some people say they're a biological agent, some people say they're a chemical agent. There are 2 toxins that are specifically listed in the Chemical Weapons Convention, ricin and saxitoxin. And so that's why the CWC and OPCW thus has an interest in being able to detect these compounds. So in that case of those toxins, it's the Forensic Science Center and DevCom CBC that can analyze those samples for OPCW. So when we're analyzing samples, whether it's a chemical warfare agent or really any WMD, there's many things that we're thinking about. The first and foremost, we're looking at what is the matrix? What is the sample you're actually sending us? So that can really affect some of the analyses that we do. If it's a liquid, we're worrying, is it aqueous or is it organic? If it's a soil, we're worrying about, the pH of the soil, of the humic content, and sometimes if it's a commercial material, we're worrying about, the solubility and, what's it treated with? Is it permeable to any of our solvents? And we really kind of think about what is actually coming in the door and what does that mean we need to do with it. Part of that is the safety aspect. So if we're getting a human blood sample, that needs to be handled in a biosafety level 2 facility to make sure that we're protecting ourselves from any contaminants that may be in the blood, as well as protecting the sample from anything that we may put in there. Depending on those matrices, it depends what we're looking for too. So if it's a biomedical sample, we may be looking for metabolites. If it's an organic or a water sample, we may be looking more for some hydrolysis products. We really try to target our analyses based on what we can know about the sample when it comes in the door. We worry about how it was collected. Did they have a nice sampling kit? Did they use clean vials, clean spatulas? Did they have chain of custody? We hope the answer to all these things is yes, but oftentimes it's not. Oftentimes it's a Ziploc bag full of, a grab sample that we may or may not, have a background or a baseline sample for. It may not have been refrigerated, so we don't know, how long it's been at what temperature. So all of these things, we would like to have as much information as we can when a sample comes in so that we can do the best analyses that's possible. And I think a lot of that comes with experience and working with these types of materials for a long, long time. We've identified what a lot of these tripping points could be and hope to address those early on in the analysis so that we don't waste sample and then realize we should have done it differently, or we wish we would have done it differently. I will also call out, we are very lucky to have a lot of advanced analytical instrumentation. The better your instrumentation, the better your detection limits are going to be. Usually that's a good thing. Sometimes it creates a whole lot of data that you have to comb through because you can detect everything in a sample, not just the target chemicals that you're looking for. And that really kind of comes back to this idea of integrating the R&D and sample analysis in a single facility can really help understand how these things matter and do things right the first time and not have to go back and repeat them. As a designated laboratory, OPCW requires testing. And we have to take an annual proficiency test for each of the matrices that we're accredited for. So we do one for environmental samples, one for biomedical samples, and we do a confidence building exercise for toxin materials as well. And how they run these is that one laboratory volunteers to make the samples, one laboratory volunteers to grade the reports, and everyone else takes the test. So you'll receive a blind sample set. You get 6 samples. 2 matrices. So if it's, say, a soil and a organic liquid, you get 3 soil samples, 3 liquid samples. One would be a control, which the laboratory would have spiked a higher concentration of a chemical in there. One will be a blank with nothing in it. And then the other would be the authentic sample that could have nothing in it, or it could have 10 things in it. You really don't know, and you don't know which is which. And for the moment we get those samples, we have 15 days to do the analysis and provide our report. I should also mention that all of our work for OPCW has to be done under an ISO 17025 certification to ensure that all of our data and results would be legally defensible. Creates a lot of additional paperwork that has to make sure that we know exactly where every microliter of a sample has been and what has been used for and can follow them cradle to grave at any point in the process. So there's an example here of some of the kinds of samples that we get. You know, I mentioned we could get, you know, like a piece of metal. This was to simulate like a blast fragment, a piece of ammunition. So, you know, when you get a little square of metal, we spend a lot of time thinking, should I cut it in half? Like, what if I put an aqueous solvent on it? Now I change the chemicals and I cause things to hydrolyze. But if I cut it in half, what if the chemicals are only on half the sample and the half that I do my extractions on didn't have any chemicals on it? And so, you know, we put it under the microscope and we see, oh gosh, it's got this pitting, and we can see some of these stains, and we can spend a lot of time trying to figure out how to handle a sample. But you know, that's kind of what the job is, right? And that's where it's looking at that experience and doing what do you think is best for each sample to maintain all the information. We get some emulsions, or we may get something that looks like it's a homogeneous mixture, but then you shake it up and you can see there's a surfactant in it. That changes how we process the sample. Soils are always complex. Soil is not soil is not soil. And so every time we get a soil, we always wonder what is its composition? How is that going to affect what I'm doing? There's always contaminants. Something like diesel fuel can really complicate an analysis. Or we'll get nice things like this, which is just an auto sampler vial that was snatched from a laboratory, where we're looking at just this little bit of trace material in the bottom. You can see it's orange in color. That dye is another chemical that's going to confound our analyses. So we have a lot of fun when we're doing these samples. It's actually one of the only times that our whole team of about 22 people are working on one thing together all day, every day for 15 days. So it's challenging, it's stressful, but it's also very fun. And here's an idea of what we do with those samples. So if you take a single sample, we end up processing it in many different ways. So if we can split it, we will take small aliquots of it. We will do some organic extractions, some aqueous extractions. We'll do some reactions to derivatize the chemicals so they can go through some of our instrumentation better. We will try many different solvents, and you see all the white boxes here are all the different instruments that we may use. Every sample goes on multiple instruments. And so when you take one sample, you prep it, 10 different ways, you put each of those 10 samples on 4 different instruments. We're quickly creating a lot of data, and all of that, of course, with chain of custody to maintain our ISO, we end up with a lot of data, a lot of different pieces of data here that we then try to put together and figure out what we think is in the sample. We use a lot of databases. We do a lot of manual structural elucidation to figure out what a compound is. And we come up with, okay, I think this is the structure. I think this is what the compound is that's in that sample. But we don't stop there because we want to be sure. So we turn to our synthetic chemists, which are on our team as well, and say, hey, can you make this? And you give them usually a Post-it note with the structure scribbled on it, say, hey, can you make this? And they come back a day later and say, here you go. So we'll analyze the standard that they made in the same ways we did the samples and make sure everything looks the same. If it does, then we're confident in our identification. If it doesn't, we're back to the drawing board to figure out again, what is that structure? So a lot of work goes into each compound that we identify. And then at the end of the day, in this case, we figured out that this is the compound. Then we look at the CWC lists. This is not reportable. It's not related to chemical weapons at all. So we can't report this in our report that formally goes to OPCW, or we would fail. If you have a false positive, if you report a chemical that's either not in the sample or not relevant to the Chemical Weapons Convention, you fail the test and you will get suspended or lose your designation. On the other hand, though, if you have a false negative, so if you miss a compound and don't report something that is in a sample, you drop a letter grade. And you have to maintain a rolling average of 2 A's and a B to maintain your designation. So if you miss 2 compounds, you have a C and you're suspended. So it's high pressure to get every one of these right, which is why we put so much time into it. In a way, we do the same thing with the biomedical samples. Shown here are some plasma and urine samples. That's typically what ends up being collected because it's relatively non-invasive. We look for small molecules. We're looking for metabolites, particularly in the urine. In the blood, we can look for large molecules as well. There's a lot of protein in your blood. So a lot of these chemicals will bind to those proteins. And now you have an adducted protein molecule. That's our target chemical. The good thing about those is that they tend to have a longer lifetime in the body. So if it's been, a month before you have access to a patient to collect a blood sample, these large molecules tend to stick around that long and can actually tell us what may have happened, up to maybe 3 months ago. The test is run in the same way as a round robin with a control, a blank, and a authentic sample with that 15-day time period to get reports in. So, I've used kind of the example here of talking about CW and OPCW, the work we do for OPCW as an example, but really no matter where a sample comes from, we're really thinking about these same issues and we go through the same process. I have some of our main partners or sponsors that we work with listed up here. I've mentioned the FBI, DOD, of course, and other US government agencies are big supporters of our work. And really we pull all these pieces together. And the nice thing about the working at the laboratory is that if we have a sample that has something that's beyond our expertise, we can turn to those other folks at the lab and pull them in, whether it's just for a single sample. So we say, hey, can we borrow you for 2 weeks? And then they can go back to their day job. Or sometimes it's, hey, this is going to be a continual effort. We'd like to have you working on a project with us for a year. And both of those options can work. So we can pull in those different expertise on an as needed basis, depending on what actually ends up coming through our door. I want to mention a few of the R&D efforts that we do as well. So I hope that you've seen how a lot of this analysis of these samples really relies on this base foundation that we have or an understanding of these chemicals. And how we get that really is through our R&D program. So, if we think about a scenario with a chemical coming through the door, if we're going to get a vial from a lab, or if we're going to get a piece of tile from a floor, we're going to extract those samples, put them on our instruments, get data from them, and then we'll try to use, look at the materials that are in that sample to identify the chemical. We start looking for attribution signatures. So what's the other stuff that's in the sample that can indicate what chemical was used, or how it was made, or where it was made, how sophisticated the person was that made it. Did they make it in a large scale laboratory, or is it, a dirty garage setup? And we call those chemical attribution signatures, or CAS. And that's really what we want to understand, because sometimes that can be all that remains in a sample if the agent itself has degraded. And so on the R&D side of the house, we think about that of, okay, if we get a sample, we're going to have to, analyze it, and we want these signatures to have meaning to us. So we end up kind of flipping that. And on the R&D side, we say, okay, well, let's make VX every different way till Sunday, so that we can understand what are the different signatures that are present based on those different routes. And then we'll take those materials and spike them into milk, or spike them onto carpet, or spike them into soil, and see how those signatures persist in any of those different surfaces, so that we're better prepared when a real sample like that does come through the door as well. I've mentioned our synthetic chemists a few times. We are allowed to synthesize up to 100 grams of CWC scheduled materials each year, where we have declared that is a capability that we have. So that's considered low enough level that it's not a large scale production facility. So we're able to do this work with actual agents so that we can understand all of these signatures. We're also trying to push the envelope on how you can identify that some of these materials have been used. One of the cases, or one of the materials that actually has a lot of domestic applications as well, is chlorine. So it's a toxic industrial chemical. Yes, it can be used as a chemical warfare agent, but also it's used, for a lot of things in U.S. industry. And so, there can be rail cars that are driving down the tracks, and if one of those were to tip, and you could have a chlorine release here in our own backyard as well. The problem with chlorine is that it's a gas, so it dissipates. So if you don't have access to samples pretty quickly after an exposure, you may not be able to identify the chlorine. So we started looking at plants. Plants are everywhere. Anywhere that you would think that there would be a chemical exposure like this, you're likely to find a plant. So we started to think, well, those plants are getting exposed to the chlorine gas too. What is it doing to them? Is it changing their metabolism? Is it changing, their proteins? And so we've been doing a study now looking at exactly that. So you can see in the photos here, when a plant is exposed to chlorine, you see there's this distinct tissue damage where it's kind of yellowing. And by doing a lot of our analyses here with our mass spectrometers, we have been able to identify some small molecules that are indicative of chlorine exposure, as well as some of these larger protein adducts, like I mentioned, we would find in a human. The very interesting thing about this is that when we did these studies, we were using a type of grass that grows in Syria. And one of the proteins that we see the chlorine attaching to is called rubisco, and that's present in every photosynthetic organism. So if you are a plant and photosynthesize, you have that protein. So we're very hopeful that this technology would work on any plant that's exposed to chlorine. To mention a little bit of the work we do in training, I mentioned that we do a lot of international engagement to help support first responders. This can cross the gamut from emergency response to evidence collection to forensic analysis, depending on the capabilities that they have and the training and staffing levels that they have. So this can be things as simple as, what kind of personal protective equipment or PPE should you wear? How do you put it on? How do you safely collect a sample without exposing yourself? How do you provide, first aid or medical care without exposing yourself? How do you collect a sample with clean glassware and clean spatulas and seal that appropriately, to maintain chain of custody so that you can get it back to a laboratory for analysis? You see in the middle photo here, they're using a lot of handheld detectors. So how can you get some early information that you can pass on to assist in an investigation? And then at the end of the day, we hope to be able to better their forensic capabilities as well. So when they get that sample collected back to the lab safely and can do a full forensic analysis on it. So it's been very valuable. And as you can see from the photo on the TV here, when there are these types of attacks around the world, we do need first responders in every nation that are able to respond to those situations safely. Okay, so I want to mention just one last thing on some of the traditional forensics that I mentioned. I mentioned some of this criticism that there's been, particularly on hair analysis, as being very subjective. And so we started an effort probably about 6-7 years ago at this point, looking at ways that we can make analysis of hairs more objective and more quantitative. So we started thinking back to, okay, well, what is hair? Hair is made of protein. Well, we do a lot of analysis of proteins. We start to think of, well, where does protein come from? How does your body make your hair? How does it make the protein? And your protein is made from a series of amino acids that are designed based on your DNA. So DNA is the gold standard for human identification. There's databases, it's very quantifiable, it's very objective. And the uniqueness of someone's DNA is based on small mutations or single nucleotide polymorphisms, single mutations that make their DNA unique. So if your DNA makes your protein and your DNA is unique, we said, is there a marker that those mutations would get translated into a mutation in that amino acid that makes up your protein? And it turns out that is the case. So if you have a mutation in your DNA, you get a mutation in your sequence of amino acids that makes up your protein. And that's something that we can detect with analytical instrumentation like mass spectrometry. So your hair is made of protein. With this technique, we've been able to identify individuals using just 1 inch of a single hair. Your teeth are made of protein, your bones are made of protein, your skin is made of protein. So we're able to use this technique on all these matrices. So that opens up a lot of avenues. You know, protein is very stable. Your hair, your bones, they'll be around for a long time, whereas DNA can degrade quickly if it's not stored appropriately. And so if you think about, you know, from an archaeological perspective, a skeleton could then potentially be identified based just on the bones, not relying on the DNA. If you're at a crime scene, if they're a victim and a perpetrator's blood is mixed, you can make that analysis very complicated to figure out what belongs to whom. But if you have hair or a piece of bone, it's from a single donor. So it solves that problem. So we've been looking at it for some of these applications shown here. Human identification, identification of unidentified remains has been really the push for this, and trying to get the chemistry to the point where we can say with a statistical probability equivalent to DNA that we're confident that a sample came from a certain individual. So it's been an exciting development to really try to push that traditional forensics field forward using some of this high-end analytical capability that we have. So with that, here's a photo of the team. This is our OPCW team when we took one of the tests during COVID. So you see we're all wearing masks, but it's been a fun time to be a part of this group. And with that, I'll take any questions.
How's it going? Name's Grant.
Hi.
Active Air Force guy for a lot of years now. So when I see OPCW and China and Russia and Iran and countries like that are members of it, is there any political challenges do you have when dealing with some of these organizations with countries that are like not friendly to the United States, or that you feel like maybe their scientists are influenced by outside factors that are beyond just the realm of science? In your role, do you have to fight any of those political battles at all, or is it what's it like working in an organization where those are co-signatories as opposed to like adversarial nations?
Yeah, that's a good question. So, with how OPCW is organized, their lab network is run through they have a technical secretariat. So we get protected from a lot of the politics. That's run on more of the executive council side of it. And that's where our Department of State covers those interactions. So our interactions really are scientist to scientist. And, that's the nice thing with the treaty, having kind of very specific guardrails around it. It's the conversations and discussions kind of stick to what's inside the scope. And, it's yes, that there are, other nations in the room, but at the end of the day, everyone has, particularly with the designated lab network or designated lab nations, it's chemists talking to chemists. And so we're talking about mass spectrometry and, molecule fragmentation and chemical ionization. And, we kind of can intentionally keep our heads down and out of the political part of it. But that certainly is, a big component of this other side of it as well. And, we are in close contact with, State Department to make sure, that we don't want to inadvertently create or walk into any landmines either. So to make sure that everyone is kind of aware of the discussions and what the challenges are from the science side as well.
Does your lab send people into zones like Syria to collect samples, or are you relying on people on the ground there to collect and then get you those to look at?
Yeah, so OPCW has their own collectors. So it's an international organization. We don't have any. We as Livermore don't have any. But those inspectors they'll rotate through and will go, based on what's happening in the world, or if they're doing, a lot of it with surveying the destruction activities. They kind of rotate through, and it's all kind of UN owned equipment that they'll use in all their PPE and everything. So they're on different teams. So they would be the ones that would go and collect a sample. It would go to OPCW's lab first, and then they would send it out to designated laboratories. We don't do the collection.
Do you ever have any trouble getting those samples out of the countries from which they're derived?
I'm sure there are issues. How OPCW works is that typically a signatory nation has to ask for assistance in order for the inspectors to be allowed to go in. So OPCW isn't just going to thrust itself upon a scenario. So someone has asked for them to come in to help. And so, they then help the OPCW get the samples out.
Thank you.
And with regards to this week in the news, if Putin were to use a nuclear weapon or chemical weapons, how would that affect your organization? What would happen? What are your thoughts?
So I'll take it from the chemical side first. So, if a chemical weapon were to be used and OPCW were to collect a sample, if they were to ask us to be one of the 2 designated laboratories that was going to analyze those samples, then yeah, that would be putting us on high alert and trying to figure out how make sure we can get the samples to us appropriately, do those analyses. We'll make sure that we have is anyone on vacation? Do we have all the right people that we would need to do those analyses? And so, anytime we hear in the news, any of this kind of inkling of chemicals being used, we're always kind of on that high alert to hear, is OPCW going to be, collecting any samples, or who is collecting samples, and might they ask for our advice or help? On the nuclear side, I think it broadens the responsibility within, certainly within the U.S., but specifically within Lawrence Livermore. We do have a nuclear threat reduction program as well that we do work closely with. And so they would be involved in some of that as well. From a law enforcement support, the Forensic Science Center would work those issues, because we would want everything to be ISO certified if anything were going to go to court. So we would certainly be involved in those. If the laboratory was asked to help with collection, whether that is advice on collection or boots on the ground with collection, that decision is made at a much higher level. But is that within our capability? Yes, we do have people that can do that.
We're from the Vietnam era, and I was just wondering if you could tell us anything about Agent Orange.
So Agent Orange is an interesting one. It's not intentionally used as a human agent, right? It's like a defoliating material. So how they've defined chemical weapons, they're things that do harm or affect the health of humans or living organisms. And so it's always that line of like, do we consider the plant a living organism? Is that considered, a CW or not? I think in the case of Vietnam, that was pre-CWC. It was sort of during that, where the Geneva Protocol was in place. And so it didn't really fall necessarily into those formal definitions. It was certainly discussed in those initial disarmament conversations as the CW, with what kind of turned into the CWC negotiations, but it's not specifically called out right now in the CWC.
Yes, I have a question. You'd mentioned here's only 2 labs in the country who are able to, address these issues. Do you work with any of the other national laboratories as support, or do you find yourself really just those two facilities?
Yeah, so, I think with the restrictions on working with scheduled materials, Livermore really is the prime location for working with CW scheduled materials. I think when you start looking at, kind of stepping that back in the process to starting materials, then some of the other national labs do some work in that area. So, certainly Los Alamos, Sandia has done some degradation work or decontamination work. PNNL, Pacific Northwest, also does some work in this area. So we do stay in touch with them. We're part of there's a Chemical Forensics International Technical Working Group that several other labs are a part of as well, where we can discuss some of these issues and newer technologies that could be applied to this area as well. So, and certainly DevCom CBC is a part of that organization too, as well as some of the other international laboratories that work in the space as well. So yeah, we're constantly, you want to be able to validate your results and make sure that someone else can reproduce them as well. So whether that's, publishing data, publishing methods for, in journals with scientific peer review. If it's information that we want to protect, then it may be with these, kind of bilateral, multilateral conversations, or certainly discussions with the other designated laboratories to make sure that we do have that level of peer review, so that if a technique is used and was something that had to go to court, whether it was domestically or in the international court, that we have that validation that the technique is sound.
Thank you so much.
You just used the phrase, if the technique is sound. Any examples in the last few years of maybe science that was accepted 10, 20, 30 years ago, where there's been some breakthrough, some discovery where things have changed radically and you look at it and go, "Ooh, we've been doing that wrong," or we've been misunderstanding this concept?
Yeah. So when I use that term, there's evidentiary standards in the U.S. court. There's either, Daubert or Frye courts of how they define, what needs to be done in order for a technique to be introduced into court. We focus more on the Daubert standards, which require that, that the error rates are known, that it's been, and the big one is that it's accepted by your peers. And that's why you really rely on a lot of publication, because by nature of publishing in a peer-reviewed journal, your peers are reviewing the data and have had that chance to provide that feedback. And so that's where it's hard with, some of these, particularly like some of the synthesis signatures, right? If we know that there's a specific compound, if you make VX using this pathway, you end up with this impurity. And if that impurity is there, I know this is the only way you could have made that. We don't want to publish that. We want to protect that kind of information. And so that's where we struggle. If we're not going to publish it, we want to make sure that we get appropriate peer review. We want to make sure that's a signature of the method and not a signature of our synthetic chemist working in our laboratory. So those are the kinds of things where it's really valuable to have either working domestically with DevCom CBC, or we've partnered with the Swedish Research Defense Agency, which is an OPCW lab over there, where we've actually exchanged materials and methods, and so we can validate each other's results in that way. So we have these joint reports to be able to show that it's not a signature of our synthetic chemist. It is an actual signature of a method. But yeah, as far as kind of the general science being criticized, that that National Academies report that came out in 2009 really did take a hard look at a lot of that, of things that at the time were accepted, right? Like hair analysis was accepted. That was how you did it. It's always been accepted as the way you do it, the way you did it. And I think with that report, they've gone back and looked at a lot of those analyses, gone back and repeated them too, had another expert analyze samples. And when you do find inconsistencies there, that's when it is a problem. And that's really why there's been this push, a recommendation to go to more objective methods, because then, you know, you can quantify your confidence in the identification. Whereas if it's just an individual, there's a lot of nuance that goes into that you can't really capture in a report, and thus is not necessarily, not that it's inherently irreproducible, but you know, to be able to prove that it's reproducible can create that challenge.
So my question is, are you finding that most of your like forensic samples that you get in, are they international samples, or do you take a good amount of stuff from like just the US?
So we can do either. It depends on, it depends on who has the sample. So I did mention the FBI, we are a partner lab with them. We're happy to help law enforcement when asked. Usually the hard thing is figuring out you know, the financials of doing the analyses. I think that's usually the biggest struggle. You know, we are a government-owned, contractor-operated laboratory, so we're full cost recovery. So we're not allowed to, although we may want to help, we can't do it unless we're getting paid to do the work. And so in some ways that can restrict the samples that we can take, but you know, we're happy to try to find a path forward for any of the requests that would come in.
Do we have any other questions for Dr. Williams? Okay.
Great. Well, thank you all so much for coming out or for watching through Zoom, and thank you to Dr. Williams.
Thank you everyone for joining us, and Dr. Williams is available to chat. There's a little meet and greet after this, so you're more than welcome to stay and speak with Dr. Williams. And thank you all.